IB Bio, Genetic Modification and Biotechnology, Part 2, looks at techniques of gene transfer among species, and then the genetic modification of specific domestic animals and plants. The essential idea is biologists have developed techniques for the artificial manipulation of DNA, cells, and organisms. Here is an outline of the 3 parts in this biotechnology series. As you can see, Part 2, this movie, is focused on gene transfer and the specific species that have been genetically modified, known as transgenic species. Part 3 looks at how we isolate genes so that we can manipulate them, gene amplification, and the analysis of DNA information. Here are our first relevant IB syllabus statements. State that genetic modification is carried out by gene transfer between species. Outline a basic technique used for gene transfer involving plasmids, a host cell, restriction enzymes, and DNA ligase. Gene transfer begins with isolating the gene of interest. I will not cover how this is done in this movie. To know more about finding the gene of interest, see Biotechnology Part 3. But the technique of transferring genes among species involves the following things: restriction enzymes, DNA ligase, vectors such as plasmids or viruses, host cells in which to grow the DNA to amplify the gene, and a mechanism to identify the clone colony that contains the gene of interest. Gene transfer involves artificially constructed plasmids. Plasmids have become one of the most important DNA biotechnology tools. Plasmids are naturally occurring rings of DNA in the cytoplasm of bacterial cells. Plasmids contain enough DNA for a few genes, but often among the plasmid genes are those that ultimately protect the bacterium from molecules that might cause harm. Hopefully you recognize all the structures in this image. If not, you need to go back to review your knowledge of prokaryotic cells. Once again, plasmids are small rings of DNA within the cytoplasm of bacterial cells, and these are separate from the principal chromosome of the cell. Just to foreshadow the next slide, it's important for you to view plasmids as a ring of DNA, as you can see here, and at a more magnified level, a ring of base pairs, as you can see here. Here is a drawing that I've made of a plasmid showing the 2-stranded structure of DNA, but the DNA on this plasmid has been cut on both sides by a restriction enzyme, an enzyme that has a very specific cut site known as a restriction site. The restriction site on one strand is not directly across from the other, as you can see here, and thus there are sticky ends which project from each strand. The plasmid and the restriction enzyme are the first 2 of our gene technology tools used in gene transfer. Restriction enzymes are naturally occurring DNA cutting enzymes in bacteria that protect the bacterium from viral infection. In this example, the restriction enzyme is EcoR1. EcoR1 cuts the DNA at a location with a GAATC sequence, leaving the sticky ends that are identical. Here is another image showing the specific restriction site of the EcoR1 restriction enzyme. You can see the GATTC restriction site that leaves identical sticky ends. If you're wondering how all of this leads to gene transfer, be patient, pay close attention. Now imagine that the DNA holding the gene of interest is cut with the same restriction enzyme as the plasmid. Both the DNA holding the gene of interest and the plasmid have complementary sticky ends. If this piece of DNA from the plasmid is cut away by the restriction enzyme, and the gene of interest, which has also been cut by the same restriction enzyme, is introduced, the 2 different pieces of DNA from the plasmid and the gene of interest, when mixed, will hybridize. The sticky ends will anneal, will come together and form a plasmid with the DNA of interest inserted into the plasmid. DNA ligase seals the gaps on both sides of the sugar phosphate backbone of the double-stranded plasmid. DNA ligase is the third tool used in the gene transfer process. Here's another look that may help you see the process. Here's your DNA of interest, let's say from the human genome, including the gene for insulin. Here's your plasmid. Both the DNA of interest and the plasmid are cut with the same restriction enzyme, such that each piece of DNA from different species have complementary sticky ends. When mixed, the DNA of interest and the plasmid, the two different DNA molecules, will come together to form a transgenic plasmid. The DNA of interest will insert and bind to the plasmid because the sticky ends on both molecules are complementary. This is where complementary base pairing becomes so important. DNA ligase forms the covalent bonds on both strands between the nucleotides along the sugar phosphate backbones. Here is a plasmid that has had this piece of DNA, another plasmid, inserted into it. The restriction site was here, and its sticky ends are now bound to this larger plasmid here and here. These two genes, along with a gene for ampicillin resistance, have now been incorporated into this larger plasmid. Now, ampicillin will kill bacteria, but the gene for ampicillin resistance will allow any bacterial cell that has this plasmid to grow in the presence of ampicillin. In other words, ampicillin resistance will allow scientists to identify bacteria with the functional plasmid, with these genes in particular, because the bacterium will grow on agar with ampicillin embedded into the agar. This image of a plasmid comes from a commercial biotechnology company. The PBLU plasmid is one we might work with in class. You can see the gene for ampicillin resistance, as well as another gene for beta-galactosidase. The restriction sites where certain enzymes cut DNA are labeled with letter-number combinations. You can see the ECO-R1 cut site here. HindD3 will be another restriction enzyme that we might use in class. Here is an electron micrograph of a plasmid. You can clearly see it as a complete ring. Remember that DNA from different species can be joined because the genetic code is universal. So when genes are transferred among species, different species, the amino acid sequence of the polypeptide will be unchanged. So let's use a bit of review to jump forward in the description of the technique that transfers genes between species. A plasmid is extracted from a bacterium and cut with a restriction enzyme. DNA of interest is extracted from another species, such as a human, also cut with the same restriction enzyme. The two pieces of DNA have complementary sticky ends because of the nature of the cut made by the restriction enzyme. The two pieces of DNA are mixed with DNA ligase, and a recombinant plasmid is formed. The recombinant plasmid is then inserted into another bacterial cell called the host cell. While you do not need to know the details of how bacterial cells take up a plasmid, we might be conducting exactly this step in class. Then the cells are cultured and allowed to proliferate, grow in number, and only the cells that have taken up the plasmid will survive on the ampicillin medium on which they are grown. And with more and more host cells dividing by binary fission, the gene of interest is also increased. The gene of interest is cloned. Now let me take a step back. I've simplified the process of getting the gene of interest. Truthfully, if the gene of interest is the gene for insulin, no one extracts it directly from the cell of a human. Instead, restriction enzymes are applied to the entire human genome. Many DNA fragments are formed, most of which do not have the insulin gene, but some that do. When these fragments are mixed with plasmids, both of which have been cut by the same restriction enzyme, most plasmids do not have the insulin gene, but some do. When plasmids are inserted into bacterial cells, most of the cells do not contain the insulin gene, but some do. Here is an image that shows the use of a plasmid to transfer a gene into a plant. Here's our gene of interest. Here's the plasmid with its restriction site. Here's the recombinant plasmid, and we can see that the plasmid has been inserted into a plant cell. Then the plant expresses the trait of the gene because when genes are transferred between species, the amino acid sequence of the polypeptide is unchanged because the genetic code is universal. So here's an image showing the same process that I've previously described, except we're using a fruit fly. We extract the DNA from the fruit fly. We extract the vector, the plasmid, from bacteria. We cut the DNA from the fruit fly using a restriction enzyme and the same enzyme to open up the plasmid, the vector. We create the recombinant DNA. We introduce the recombinant plasmid into bacteria. We allow them to proliferate, and we select the clone that has the gene of interest. Now, there's a lot of detail in selecting the clone with the gene of interest, but marker genes, such as ampicillin resistance, allows us to see which bacteria have taken up the plasmid. That's a first step. There'll be more on this in part 3. Once the recombinant plasmid is created and inserted into a host cell bacterium, and the host cells increase in number, and the colony with the gene of interest has been identified, the bacteria are held within a culture, a fermentation tank, where the insulin is made in abundance. Just as a note, it's important for me to say that a virus, when mixed with a gene of interest, seen here, can serve as the vector for delivering new DNA into a cell. And here are the next important IB syllabus statements for this movie. State 2 examples of the current uses of genetically modified crops or animals. Discuss the potential risks and benefits associated with genetic modification of crops, and analyze data on the risks to monarch butterflies of Bt crops. Bt corn was genetically engineered with a bacterial gene that allows the plant to resist insect pests by making a molecule that kills insects. You can see in the image the upper ear is Bt corn. It's a genetically modified corn plant with the Bt gene, allowing the plant to resist insects that would eat the corn. The lower ear is from a traditional variety without the Bt gene. Now, the Bt gene, this corn, would allow farmers not only to have less crop loss, but farmers don't need to spray as much pesticide on the field. There are 2 advantages. But here are the concerns. Does the genetically modified crop kill unintended insects? Is the Bt molecule/pesticide harmful to people? Does the gene get into other plant species that suddenly no longer have a biological control, and thus they spread rapidly like an invasive species might? One advantage of genetically modified crops, such as Bt corn, is that the use of pesticides on agricultural fields would decrease. You can see in this graph that as Bt corn planting has increased since 1996, insecticide use has decreased. But insects other than the corn borer, such as the monarch butterfly, have become unintended targets. And the mean survival rate of monarch larvae exposed to Bt pollen declines relative to controls. Here you can see that larvae feeding from leaves exposed to Bt pollen have a lower survival rate than larvae feeding on leaves with no Bt pollen. Since 1994, the number of overwintering butterflies in Mexico has generally decreased. Now, the explanation for the monarch decline in Mexico may well be Bt crops, but we must be cautious in our conclusion. The monarch decline could also be habitat loss, as a herbicide known as glyphosate reduces the frequency of milkweed plants, and milkweed plants are a natural habitat for monarch butterflies. In assessing ecological risks, genetically modified crops present two types of hazards. There is the exposure hazard, which includes pollen-mediated gene flow, plants escaping cultivation, and plant material into the environment. The ecological hazards would be gene flow to related species, non-target effects, and increased plant weediness. Let me move away from Bt crops and provide you with a second example of transgenic crops. And I'm going to start the story with Roundup. Roundup, also known as glyphosate, is a herbicide. It kills plants by interfering with amino acid synthesis, specifically phenylalanine, tryptophan, and tyrosine, all essential amino acids. Now, Monsanto has engineered Roundup-ready plants that can survive the spraying of Roundup. Roundup-ready soybean is just one example. Now, Roundup-ready plants allows farmers to spray Roundup liberally, it's a herbicide, to kill weeds that would compete with the crop. Now, this would increase the farmer's yield, but it also allows farmers to spray Roundup liberally on the field without worrying about their crop, and this increases the amount of this herbicide in the environment. Now, is agriculture controlled by corporations that patent Roundup-ready seeds? Is the genetic diversity of our food supply at risk? And will the gene be crossed into other species, creating superweeds that can proliferate? But a downside of growing crops that are resistant to an herbicide is that farmers can use more of the herbicide to kill off the weeds. Here is a graph that shows the rate of glyphosate use, this is the herbicide, since the introduction of Roundup-tolerant plants. The graph suggests a positive correlation between the use of herbicides, glyphosate, and the introduction of genetically modified Roundup-tolerant crops. You can see in this graph that the use of genetically modified crops, herbicide-tolerant soybeans, herbicide-tolerant cotton, Bt corn, has generally increased since 1996. But so has the incidence of species resistant to herbicides increased over time. Weedy species resistant to herbicides, is a risk of using crops genetically modified to resist Roundup, a herbicide. I would encourage you to stop the movie and take the time to study this slide carefully, but right now I'd like to highlight a few items. Transgenic plants aim to create higher yields in a world with increasing numbers of people. But the concerns are the genetically modified foods safe for people to eat? Are the seeds affordable for poor farmers in developing regions of the world? And is the agricultural system controlled by Western corporate interests? And what about the natural diversity of the world's food organisms? Golden rice is genetically engineered to synthesize an excess of a vitamin A precursor. It's designed to be grown and consumed in areas with a shortage of dietary vitamin A, primarily in less developed regions of the world. Hopefully you can see the health advantages of the humanitarian effort to produce this transgenic plant. Now, controversy has followed the production of golden rice because of those who resist the concept of genetically modified foods. Opponents to genetically modified foods raise the following concerns. 1, loss of biodiversity if the world were to become dependent on a handful of genetically engineered species. 2, the corporate control of agriculture, including the high expense of genetically modified seeds for farmers, which this particularly impacts farmers in less developed countries. And 3, the potential dangers of eating transgenic foods, specifically health concerns for which there is little evidence. The Flavor Saver tomato was the first commercially produced genetically engineered food. A gene was inserted to interfere with the production of an enzyme that softened the fruit. The thinking was that the tomato could be left on the shelf longer without rotting, but it didn't go as expected. The public thought the fruit was bland in its taste. Genetic modification changes the essence of the wild crop, the taste, the texture, the color. Vitamin A-enriched rice is wonderful for people in regions of the world where there are vitamin deficiencies. But what are the risks? Are there health risks, loss of biodiversity, corporate control of seeds? Study this slide to strengthen your understanding of the advantages and disadvantages of genetically modified crops. In order to create transgenic animals, the gene must be inserted into the embryo so that all cells of the animal following development would carry the gene. You can see here DNA is being inserted just after fertilization. Again, with transgenic animals, in this case pigs that produce a leaner muscle, the gene must be inserted into the embryos. These sheep have a gene for human blood protein, factor IX, important in blood clotting. The protein is secreted in the milk of the animal and can be extracted from the milk. The bull seen in this image is a transgenic species with a gene for human lactoferrin, HLF. The bull's offspring make HLF, which will be harvested from the milk. Lactoferrin is one of the components of the immune system of the body. It has antimicrobial activity, in particular antibacterial activity for human infants. The genetically modified Atlantic salmon is engineered with genes from an Arctic pout and a Pacific Chinook salmon. These genes enable this fish to grow year-round instead of only during the spring and the summer. And the purpose of the genetic engineering is to increase the speed at which the fish grows without affecting its ultimate size or other qualities. The fish grows to market size in 16 to 18 months rather than 3 years. The concern is that the genetically modified salmon would escape into the wild and outcompete native species, if not driving native species to extinction. In this slide, genetically modified animals provide the examples. In the case of clotting factor IX, typically secreted in the mammary glands, would the gene be expressed where it's not supposed to be expressed, in tissues other than the mammary glands? Are any sheep harmed, or do any sheep die in the process of producing transgenic animals? Faster-growing salmon is terrific for providing food in a hungry world, but what if the fish escape to the wild? What happens to native species? What happens to the diversity in nature? In this example, we have transgenic flowers. Flowers that have an inserted gene, a gene that allows the flower to be insensitive to a plant hormone known as ethylene. By being insensitive to ethylene, the plant does not wilt. Here's a table of medically important proteins produced by genetically modified bacteria. We have factor VIII, which is for treating hemophilia. It's a blood clotting protein. We have growth hormone, and we have insulin. Genetically modified foods have become controversial for various reasons. One, the suspicion about Western corporate intentions juxtaposed against poor farmers in developing nations. 2, health safety. 3, the loss of natural genetic diversity within our agricultural system. And lastly, people in developed countries want their foods to be labeled clearly for what they are, while food-producing companies seem reluctant to label. But genetically modified foods might be a solution for increasing the world's food supply in the face of increasing numbers of people as the world's population grows from 7 billion plus towards 8 billion. Protests around the world have been common, although resistance to genetically modified foods is more about what we don't know rather than substantiated concerns. And that brings us to the end of IB Bio, Genetic Modification and Biotechnology Part 2. In Part 3, we will focus on isolating the genes of interest, gene amplification, and DNA analysis.